4, 3, 2, 1.
Hello, everyone, and welcome to today's keynote webinar titled Advancing Toward Recovery from Post-Acute Sequelae of SARS-CoV-2 Infection, the NIH RECOVER Initiative. This webinar is part of the 8th Coronavirus Virtual Event Series, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind everyone that this event is interactive and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located on the far left of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you have any trouble seeing or hearing this presentation, just click on that Help Desk button located at the top of your screen within the navigation bar from the lobby. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the continuing education credits link located in the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Walter Koroshetz, the director of the National Institute of Neurological Disorders and Stroke at the National Institute of Health. For a complete biography of our speakers, please visit the Presenter tab from the menu at the left of your screen. Dr. Koroshetz, welcome. You may now begin your presentation, sir.
Well, thank you very much. It's a pleasure to talk to you today about the consequences of the pandemic, the health consequences going forward and into the future. I'm sure everyone wishes that the pandemic would be over and things would then be perfectly normal again. Unfortunately, for a lot of reasons, that's unlikely to happen. And one of those reasons is that people are still suffering persistent symptoms even after the acute infection is over. And so that's what we're going to be talking about today. And so I run the Neurologic Institute here at NIH. I'm a neurologist, and I've been involved in with NIAID, the Infectious Disease Institute, in running our work in chronic fatigue syndrome, myalgic encephalomyelitis, chronic fatigue syndrome, which is another post-viral illness that is very similar to what we'll be talking about today. We've been doing that for about 10 years now. Very little progress, unfortunately. Now, the RECOVER Initiative, I'm helping to run with Dr. Gary Gibbons and Amy Patterson from the Heart, Lung, and Blood Institute. And I'm going to talk to you today about what we know and mostly what we don't know about this these post-acute sequelae of SARS-CoV-2 infection. I guess what we do know is that the numbers are pretty significant. Depending on the study population, whether it's been hospitalized, non-hospitalized, whether it was Omicron or Delta variant, the numbers vary, but beginning studies were showing that about 79% of hospitalized people were still having trouble, and at the 3-month point and at 6 months, about 55% still having sequelae of COVID-19 infections. And from this publication, you can see the type of things that people were experiencing. The point to make is that they're experiencing multiple symptoms, not just one. On average, people have clusters of about 7 different symptoms. And you see here, some of these are neurologic, headache, taste disorder, smell disorder, cognitive impairment is going to be a big one we'll talk about, memory deficits, difficulty concentrating, particularly with respiratory infections in the hospitalized patients, lung fibrosis, restrictive pattern on spirometry, persistent cough, dyspnea, shortness of breath, even with normal pulmonary function tests, abnormal pulmonary function tests. These are all pretty common. and these affect people's mobility. There are also cardiovascular symptoms like palpitations and chest pain. And there's also serious mental health aspects to COVID-19. Some of these may be due to the virus. Some of these are due to the fact that, the pandemic itself took a toll on members of your family, your friends, and you were then at risk. But depression, sleep disorder, anxiety disorder, post-traumatic stress disorder, very common. Some digestive disease problems, and some of these symptoms that fall into what we call general symptoms, weight loss, myalgias, general pain, not much fever, a couple, small percentage of people complaining of fever. Fatigue, of course, is a big one. And so this led to the idea of what's called long COVID, which are these persistent symptoms, and long COVID consists of a cluster of the symptoms that we just went over. Now, what is the incidence and prevalence? The best data I can find is from the UK Office for National Statistics. And so a couple of points we made that depending on how you ask the question and what the population is, you'll get a slightly different answer. So what they have republished is that persistent symptoms after 12 weeks of acute infection are about 3% if you base it on tracking specific symptoms. If you base it on self-classification by patients, it's up as high as almost 12%. Among study participants with COVID who had COVID-19, 5% of them reported any of the 12 common symptoms. We went through a whole bunch of symptoms in the previous slides. The most common ones, the most common 12 ones, 5% report. But if you look in the general population who didn't have COVID, the prevalence of many of those symptoms is 3.4%. And that's because the symptoms that we just went through are extremely common for a whole host of different reasons in the general population. You get more specific if you look and find out who has developed these symptoms around the time of an infection and lasted for a continuous period of at least 12 weeks from infection. You compare that with a control group who had these symptoms 12 continuous weeks, and then you can see the bigger difference. 3% in those who had continuous symptoms after COVID, and 0.5% in the control group. If you go back to self-reported, it's 11.7%. And of those people who self-report trouble, it's about 7.5% claim that it results in limitation in day-to-day activities. And this goes higher if you only consider participants who are symptomatic at the acute phase of the infection. The prevalence rates were highest in females, adults aged 50 to 69, people with a pre-existing health condition, and those with some signs of high viral load at the time of infection. This is a recent paper that just came out looking at 55 different studies, I think 1.2 million people included in them. And these are the proportion of people with long COVID after COVID infection. And you can see in males aged less than 20, it's running, less than 3%, higher in men aged greater than 20. The symptoms, fatigue is common, and respiratory trouble is the next most common. And you can also see that the symptoms are more prevalent in women aged greater than 20, and that's the highest risk group. I'm not exactly sure how where these figures came from looking at the duration, but what they published is that the estimated mean long COVID symptom cluster duration was 9 months in people who were hospitalized and 4 months among non-hospitalized individuals. I think the length of time that people suffer is still an open question, and what percent go on to really a chronic disease that lasts years versus something that gets better over a year is still unknown, but this is quite promising. And most of the people we talk to do say that patients are still recovering over time. The change in the variant of the virus has also affected your chances of developing long COVID, as has vaccination. So with only double vaccination, the Delta variant had the high percentage, almost 9% of people developing post-COVID. Once you get triple vaccinated, really almost no matter what the variant, it's down 4 to 5%. So vaccination is clearly reducing your chance of getting long COVID. And also the data is that with Omicron, there's lower chance of getting long COVID. Now, besides the long COVID, which is these clusters of symptoms, we have to really be careful and watch out for what COVID has done to your risk of developing more common illnesses. So this is a little bit difficult to see, but this is looking at people less than 64 in the light blue and greater than 65 in the dark blue. And these are things like cardiac, infarction, gastrointestinal disease, acute pulmonary emboli. So these are not long COVID, but you can see that the hazard ratio for developing some of these quite serious conditions also seems to have been increased in people who had COVID previously. So there's two areas we need to concentrate on. One is understanding long COVID, and the other is understanding what COVID is doing to the common diseases and your risk for developing them in the future if you have had COVID. Again, this is from the VA data, really good electronic health record data. And I apologize for the colors. I didn't make this, there was a problem switching slides here, but in the green is the hazard ratio if you were never hospitalized but had COVID. And the red is hospitalized but not in an intensive care unit. And the really dark blue is intensive care unit. So for many of these conditions, all of these conditions actually, the risk goes up the sicker you are, highest in the non-ICU patients. But the key thing for each of these, stroke, TIA, atrial fibrillation, sinus tachycardia, myocardial infarction, the risks, even in non-hospitalized patients, do look like they've been increased by having had COVID in the past. From the neurologic standpoint, and what we'll talk about is this is one of the most disabling symptoms to people, is trouble with their quote-unquote thinking. And this has been given the name brain fog by patients, people suffering with long COVID. And studies, this was a study out of the UK where 81,000 people were part of a study. About 13,000 actually had COVID, and they were able to look at the effect of COVID on multiple different cognitive tests that could be done over the web. And what they find is that having had COVID, particularly if you had severe COVID with respiratory symptoms that got you in the hospital or got you on a ventilator, you were more likely to have trouble with cognition. But even in people who never went in the hospital, there's a significant loss of cognitive abilities in this large population study. It's more prominent for those complex tasks that require reasoning, planning, and problem solving. And the simpler functions such as word, memory span, emotional processing seem to be unscathed. But this is what the people are complaining of, is trying to get complex mental tasks done. Many of them can go to work, but it's just so much harder to get through the day managing their mental work. Now, what's causing this? And the answer is we don't really know. unfortunately, but there are a couple of hypothesized causes that have risen to the top. One of which we're talking about is persistence of the virus in the body somewhere that's causing consistent stimulation of the immune system. This could be actual replicating virus, or it could be antigens from the viral infection that are still being leaked out and presented to the immune system. The other one, we know COVID has a tremendous ability to upset your immune system. And so one other hypothesis is that the immune system never really comes back to its normal set point, and it's still dysregulated in some people, causing their symptoms. Because as you can see from what we talked to you about, many of these symptoms are pretty much what you get when you get a viral infection. The other issue, which may portend more value in understanding conditions such as heart attack and stroke, is how the virus actually damaged organs and tissues that will then in turn result in, say, accelerated atherosclerosis or worsened kidney disease, worsened diabetes, for instance. There have been some studies looking at the data that was collected in the acute stage of the illness and trying to correlate that with what was your risk of developing long COVID 3 months later. So here's a paper, and you can see what their symptoms, what the patient's symptoms were at 3 months. So fatigue, cough, trouble with smell and taste, headache, difficulty concentrating. And what they found was that people who had gastrointestinal problems, they thought they saw a specific cytomegalovirus specific CD8 T cell exhibiting unique dynamics during the recovery period. What they found is that any immunologic associations with PASC, they would see the association acutely with developing PASC later, but the later you look, the less you see in terms of an association, and emphasize the importance of looking early on. So in the RECOVER study, which we're talking about, we have a big premium on studying people during their acute infection and right around that time to see what happened in the acute event that portends having trouble coming on later on. There's another study showing that immunoglobulin 3, IgG3 levels increase in COVID. What they found was that the people who developed long COVID actually had less of an increase in IG3 during their acute infection. So that portended developing post-acute or long COVID over time. Here's a smaller study, 189 people, and this was done at NIH, where they did a lot of the kind of standard testing, not a lot of the sophisticated immunologic analysis, but they did look at things like cytokines and stuff. And what they reported is they really couldn't find abnormalities on exam. The antibody levels were highly variable, didn't correlate with developing long COVID. They couldn't find any evidence of persistent viremia in the blood. And so they really kind of closed the study saying we tried, we didn't really see the cause of the problem, which means it's actually going to be a hard problem, I think, to fix. There are a number of studies that have come out showing abnormalities in people who have long COVID. So this is now samples taken in people months after their acute infection. So in this paper, they're claiming elevated interferon beta and interferon gamma 1, persistently high at 8 months after infection. Here's the interferon beta. Red is the people who have long COVID, blue is the controls. And you can see there's actually quite a lot of overlap. But again, they did see a population change in these levels. Here's another paper. I think this one came out of Germany, 258 people 8 months after mostly mild infection. 40% of the patients still had some symptom at 6 months. So they find antibodies pretty commonly in people after COVID, self-antibodies, autoantibodies, but they don't see any difference in the titers in people who have long COVID versus no long COVID. So I think one thing to take home is that, yeah, COVID is going to cause autoantibodies. Knowing whether there's a pathogenic autobody in that mix is going to be a challenge, but maybe potentially worth figuring out. They did see correlations with TNF alpha, IL-1 beta, and IL-6 plasma levels at 8 months, more common in people who had long COVID. And their thought was that this was COVID causing inflammation in the lungs and creating pro-inflammatory macrophages that continue out in the bloodstream even 8 months later. Okay. Let me know if you didn't see the slide move. My presentation buttons just disappeared. This is a study. Oh, we're good. Okay, great. There's another study that's still in medRxiv, so it's not been peer reviewed yet, but they looked at a whole bunch of factors, some of which they saw were higher in people with long COVID. Some of them they found were lower in people with long COVID compared to controls which had COVID but not long COVID. In this group, what they see is that low cortisol was a major factor in the people who had long COVID. Also, some antibodies to Epstein-Barr virus and HSV1 and CD8 cells responsive to Epstein-Barr virus, and a number of complement and subsets of T cells were higher in people who had long COVID. Okay, there's another study looking at, again, antibodies to Epstein-Barr virus. So when you have an acute infection and you're over age 20, you're likely to get a reactivation of Epstein-Barr virus during your infection. And that, of course, is a potential culprit, as Epstein-Barr virus causes infectious mononucleosis, which cause a lot of the symptoms of long COVID, and also people with chronic fatigue syndrome. One of the major causes happens to be Epstein-Barr virus. So whether or not reactivation of Epstein-Barr virus is the culprit here, there is some evidence for that going forward, but we'll have, again, it's one of many culprits to consider. Autoimmunity we talked about. So there are a number of small studies suggesting autoimmunity in people with PASC. We do know that a lot of antibodies against self-antigens are generated during the acute infection. This one is a paper, I think this one is, yes, this one is published now in Molecular Medicine. And what they found is that there are certain vascular factors, angiotensin 1, P-selectin, MMP1, that are elevated in people with long COVID, and believe that there's some angiogenesis as a key pathologic mechanism in long COVID. We do know that the virus has a predilection for infecting endothelial cells, and so effects on angiogenesis would kind of follow from that. Again, we have how this actually creates the symptoms that people are suffering from, not as apparent as, say, it would be for reactivation of ABV or some persistent virus. In terms of the neurologic system, there have been some studies, very small studies, of one study showing oligoclonal bands. So these are antibody production in the CSF in people who have cognitive trouble after COVID. Another study showing mitochondrial proteins and also COVID-19 antigens and exosomes in circulating blood from people who have cognitive trouble.
Hey, Walter?
Yeah.
Can you click on the slide that you're currently on? Because the slide stopped advancing. Can you click on one that you're on and then select the push to audience button?
Push the audience button. Okay.
Perfect.
Got
I'll
it.
keep going.
Okay.
Thank you.
Okay. Sorry about that.
No worries.
Okay. Study of small fiber neuropathy, which again, people think may be autoimmune mediated in people with post-COVID. There's a study in animals showing that mild COVID in mice that have the human ACE receptor have problems with myelination in the brain and with neurogenesis in the brain. Whether this occurs in people, again, we don't know. There is one biomarker that was shared between the CCL11, which was shared between the animals and the humans. But again, right now it's in the hypothesis stage. Okay. This is a study looking at brain morphology over time, claiming to see changes in brain structure, mostly thinning of brain regions that are connected to the olfactory system after COVID. And we know that COVID does affect the olfactory system. There's some question of whether it gets into the brain through the olfactory nerve. But you have to be careful, because before COVID, there was a study of people who lost their sense of smell for lots of different reasons, and you see very similar changes. So it could be that what was previously reported related to the virus may just be a reaction to the loss of olfactory input, which also changes morphology in the brain. So causation is clearly, you have to be careful about ascribing causation. The next area I wanted to talk about is the idea of potential persistent virus, replicating virus potentially in the body. HIV is a great example. EBV is a great example. You get infected, the virus is never really cleared. The first paper came out from Michelle Nussenzweig in 2021, where they found that the immune reaction looks like it was still evolving, indicating that there was still some stimulation of the immune system by the virus. And then they found evidence of SARS-CoV-2 nucleic acids and immunoreactivity in the small bowel of patients who had previously had COVID. There is a paper coming out of the NIH. It's currently, again, not peer-reviewed, but these are autopsy studies on 44 people with infection over 7 months. And they're claiming that the virus is widely distributed, even in patients who died with asymptomatic or mild infection. They're claiming that they were able to prove viral replication in multiple pulmonary and extrapulmonary tissues and RNA in multiple anatomic sites for up to 230 days after symptom onset. They don't see a lot of inflammation or viral cytopathology outside the lung. And of course, this is has to be replicated, but would certainly be a big concern if there's in certain people continuous viral replication accounting for patient symptoms. And so this would have to be tracked down, both whether it's occurring and also whether it's causative. There is a report of spike antigen from the virus in circulating bloodstream from David Walt's group at the Brigham, and they're seeing it in people who have post-acute sequelae of COVID in red, but not in people who had a fine recovery after COVID. So we're trying to replicate this now in the RECOVER program, but again, early days, but something like this would be incredibly important as a potential diagnostic test, maybe as a biomarker trying to obliterate this if it's due to viral persistence with antivirals, or if it's due to persistent release of antigen from the prior acute infection, potentially with immune modulators that block the immune response to the continuous stimulation from this protein. Again, these are just some vague leads that we have now, but it's important to follow these through. With regard to that, there's also a paper that came out of a group in Kansas that is again showing in exosomes, spike protein in the exosomes from people who do have post-acute sequelae of COVID, long COVID, but not in people who had COVID and don't have symptoms. But again, these are very small numbers at this point. So now I'm going to jump to the RECOVER initiative, and this is an initiative that was appropriated $1.1 billion by Congress about a little over a year ago to try and get to the bottom of the problem and trying to get solutions for people who are still suffering. It's part of a bigger push on the part of the president to mount a response to the long COVID aspects of the pandemic. So the idea is, the plan is to understand how to predict, treat, and prevent the post-acute sequelae of COVID, and that would be the really long-term effects on, say, heart attack or stroke, but also a big concentration on long COVID and those clusters of symptoms that we talked about. So we want to understand the clinical spectrum and the biology underlying the recovery over time. Why do some people recover very well and others not so well? Define the risk factors to develop long COVID and post-acute sequelae COVID, study the pathogenesis over time, and identify interventions that would be helpful. And the program has multiple different components. I mentioned in the past that in the last couple of minutes that there's a lot of worry that this will be a very hard problem to sort out. So we put together a program that would kind of leave no stone unturned, give it our best push to understand this problem. So we have cohort studies. We're enrolling tens of thousands of people. Some of them have acute infection, and we can follow them, how they recover over time, what's the difference between someone who makes a good recovery and a poor recovery. And we're also recruiting people who are now months out and still having symptoms and comparing them to people who are months out but not having symptoms, trying to see what the differentiation is in the biology there. We have a lot of electronic health record studies going on that can look at electronic health records for 60 million people. These studies are actually have a lot of publications either already out or coming out very quickly because they have access to such amazing electronic health record data. We have autopsy studies looking to get hundreds of cases where we can look under the microscope for the kind of things we talked about: continued inflammation, changes in the immune system, persistent virus, persistent antigenemia, persistent antigen presentation by different tissues in the body. And then the hope is that we have enough evidence from these data to go into clinical trials. We also are funding studies to get at the pathobiology of long COVID, both the neurologic, sleep disorders, pain, fatigue, exercise intolerance, autonomic troubles. And we have enrolled over 10,000 people, so it's the largest in-person study of long COVID in the world. And we've funded 42 pathobiology studies many of these looking at samples that have been taken from these 10,000 people. We actually have master protocols for clinical trials. Some of the master protocols are geared to look at an intervention that would affect the disease in general and attenuate all symptoms. And some of them are based on a particular cluster of symptoms, say the sleep disorder or the pain. Some of these interventions may be actually symptomatic as opposed to getting at the underlying pathology. And then we have EHR studies that I mentioned. We have data repositories and sample repositories so that if we can't figure this out in the next couple of years, there'll be incredible clinical data matched to samples for future studies. From what we know so far is that we are getting a fairly high rate of post-acute sequelae symptoms in the patients that we've been enrolling in the acute study. And it's clearly higher in people who are hospitalized than non-hospitalized. The dominant features are basically as we expected, with cardiovascular, respiratory, musculoskeletal, neurologic, digestive, and what we call general or constitutional. We're trying to work them in, see what clusters they fall into. And we're also looking at adults as well as children. And we're also looking closely at a cohort of pregnant women who were pregnant when they developed their COVID. A number of papers have come out from electronic health record groups looking at subtypes, looking at incidence, and looking at risk of, say, cardiac complications or other complications after COVID. Well, the general kind of take-home points is that the peaks of long COVID occurred earlier in the pandemic, which were times when there was less vaccines, but also there was a more severe variant, the Delta variant. We do have evidence that vaccine protects against the cardiac complications of infections in teens and young adults, which is important because the vaccine does have, as a side effect in some small proportion, cardiac complications. And again, vaccinated continue to be at risk for post-acute COVID, but at a somewhat lower rate. Okay, I'm going to skip this one and talk a little bit about the underlying biological mechanisms, which we've mentioned before. So, consequence of acute infection, viral persistence, viral reservoirs, secondary damage, reprogramming of a host tissue organ, immune dysfunction, residual tissue injury, or changes, epigenetic changes due to COVID. So the pathobiology studies that we're funding are kind of hitting all of these general bins. And now we're working on developing these master protocols for the various symptoms, either a composite outcome where all the symptoms are followed over time, or more targeted trials at a particular symptom cluster. So some of these are symptom-based treatments that we'd be entertaining bringing into trials. Some of them are actually focused at the underlying biology. Okay. The one we're working on most urgently now is an antiviral treatment, and that's, as you can imagine, based on the data I showed you with evidence of circulating antigen, which we're now trying to replicate in our population, and also the autopsy study coming out of NIH claiming to show persistent viral replication. So the question there is, will an antiviral be able to block persistent viral replication? And would that be evidenced by a drop in the amount of circulating spike antigen and exosomes? So if everything lined up, that would be a rational way to go. And then the question is, what antiviral will work in that instance, and at what dose and what duration, and do you have to have repetitive treatments? So all these things would have to be worked out. But right now, that this seems to be rising to the top of the list of things to do, and we are currently engaged with discussing this with companies to get the antivirals into trials. These master protocol that we're developing with the antiviral therapy would also work for an immune modulator, for instance, because both antiviral and immune modulator, you would be hoping to hit a whole host of symptom clusters as opposed to one cluster at a time. So these master protocols for the antivirals and the immune modulators would look very similar. One related to sleep disorders would look something different from something related to exercise, how to improve exercise tolerance. And I'm just going to end up by saying that the RECOVER Initiative is it's really a tremendous effort on the part of tens of thousands of patients, thousands of investigators going on all over the country. It's a really heavy lift. We're really asking people to really dedicate themselves to understand this problem, and we're so grateful to the patients and to the investigators for the work. We also put on a series of seminars if people are interested in. So these are on the RECOVER website for when they're occurring. We go through a whole bunch of the different topics that would be relevant. So with that, I'd like to end, and happy to take questions. And I promise that I will not have the answers to your questions, but because there's just so much unknown about this problem. But I'll be happy to kind of engage in discussion, and maybe other people could chime in with the answers. Thank you very much.
And thank you, Dr. Koroshetz, for that informative presentation. We will now start that live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started. It looks like we already have some great questions coming in. Our first question, Dr. Koroshetz, is RECOVER planning on asking for more funding from Congress, or is it not being renewed?
So it's a good question. Turns out that NIH cannot ask Congress for money. We can present the case for what we could do if money became available. And I think that what we know so far is that the funds that we received will get us pretty far, but they will not allow us to do a lot of clinical trials. So the clinical trials are quite expensive. Some of them were costing out as $60 million a piece, for, especially if it was a trial that would go to the FDA for labeling in this condition. And so to do those scale trials, we would not have the funds.
Thank you very much. And our next question coming in, Dr. Koroshetz, at the August 26 ME/CFS telebriefing, you stated, I think ME/CFS ancillary studies will be funded by the NINDS and the NIAID. Looking at a couple now to the question of, will there be ME/CFS controls or ME/CFS ancillary studies in recovery? Is there any update on this front that you could share with us?
Yeah, no, sure thing. So I think a couple of points for people who, we mentioned it, but maybe just to expand a little bit. that, as I see it, long COVID is a post-viral syndrome, a post-infectious syndrome, not even just virus, that we have been struggling with for years and really haven't made much progress in. So post-Lyme, post-EBV, ME/CFS, these, we don't know, for sure because we haven't been able to nail it down, but the suspicion is that these are all post-viral syndromes, and the overlap between them and post-COVID is really quite striking. The one feature of ME/CFS that we're looking for in the post-COVID is something that's a little bit unusual. It's called post-exertional malaise, and that would be defined as someone who's having trouble, but on a good day, they actually do something like they go out shopping, and that basically ruins them for the next 3 days, that they're then bedbound, for instance. So that's what we call post-exertional malaise, and that seems to be a signature feature of the ME/CFS. And some people with post-COVID have had that, not everyone. But the important thing is that in ME/CFS research, we suspected it was a virus. We've never been able to find the virus. And we study people when they present with ME/CFS, which is usually years after they develop symptoms. So if you wanted to understand ME/CFS, actually having millions of people who are infected with the same virus, you know exactly when, and you can follow those people who get ME/CFS versus those who don't. This is almost like a natural experiment to understand ME/CFS. So we're very hopeful that this will lead to clues to what has been causing ME/CFS for hundreds of years probably. Now that being said, the funding that came from Congress is for COVID only. We are at NINDS, however, funding studies in which they are comparing ME/CFS with a post-COVID, particularly a group out at in Chicago, DePaul University, has been studying ME/CFS after Epstein-Barr virus, and now it's funded to look after COVID as well. If we get clues from RECOVER, I think one of the first things we'll do is to see if those clues are operational in people with ME/CFS. So that's I think the long answer, but it's a really important point that the question is making. Oh, I lost sound.
I apologize.
Can't hear.
I said thank you very much, and I want to thank our audience for these great questions as well. This next question is very related. How is RECOVER taking steps to ensure ME/CFS cases are accurately identified and tracked across these long COVID cases? And the second part of this question, how is post-exertional malaise, one of the most common symptoms being identified in long COVID, addressed in RECOVER, capturing information on triggers, severity, and frequency?
Right. So yeah, good point. And we made it a point to capture that kind of data from the beginning because of our experience with ME/CFS. So yes, post-exertional malaise is being tracked in people who are coming into the RECOVER and also longitudinally over time. And I think also we don't have exact plans yet, but interested in trying to understand how to rehabilitate people with long COVID and not create post-exertional malaise in those people. So that's a tricky needle to thread, because you want people do generally improve their exercise tolerance over time with post-COVID. But there is worry that if you overdo the exercise, then you could actually make things worse. So that's one of the kind of research topics that we're hoping to address in the RECOVER protocols related to exercise tolerance.
Thank you so much. Now, has NH asked Congress for more money to keep Recover going after 4 years? Clearly, there's a need to be an annual post-viral program at NIH.
So I missed the first part of the question about whether.
I was asking, has NIH asked Congress for more money to keep Recover going after 4 years? Clearly, this needs to be an annual post-viral program at NIH.
Right. So as I mentioned before, that we are prohibited from asking Congress for money. So NIH does not ask Congress for money. NIH can present and they can only do it when they're asked. So I just got off a briefing before on a different disease, but what's been happening is Congress asks us to give them a briefing. We tell them where we are. We tell them what the expectations are going forward, and how much those things might cost. And that's as far as we can go. So, I mean, Congress makes their decisions, and we are, we take those decisions, but we have very little influence over what Congress decides.
Thank you for
that. I guess it's the right thing.
Yes, and thank you for that. And thank you for these questions still coming in. And we have time for a couple more questions. What drug will you be trialing, and when will the trials be announced this fall?
Right. So I can't really say right now because we're still in negotiations with a number of companies. The drugs, particularly we're talking about antivirals here. There's a limited number, and hopefully that decision will be made pretty soon. But as you can imagine, it's a negotiation and an agreement, and the lawyers have to approve it. So we're still in that negotiation stage.
Thank you so much. And another great follow-up to this question, why don't you have decentralized trials with mobile labs to collect blood? Many long COVID patients are too sick to travel to the sites.
That's a good question. No, that's a good one. Yeah, that would be good to have. Yeah, that's a good idea. Now, some of this testing, well, I mean, I didn't mention this, but the recover testing is tiered. So everyone gets the same tier 1 testing, and that's pretty non-invasive, but it does require, a sophisticated investigator examining, doing tests on the person. The next level are tier 2 tests, and they're a little bit more substantial. They require a little more investment of time and money. And some of them are things like scans, which certainly could not do at home. And then there's a tier 3 testing, which is, the highest level, going and trying to understand a particular problem, whether it's a brain problem, a brain abnormality, or an immune abnormality. So I guess, reflecting on this, the way Recover is put together, the tier 2 are triggered by the symptoms. So everybody gets tier 1, but depending on your symptoms, that will trigger certain tier 2 tests. And depending on how those results go, would trigger a tier 3 test. And tier 2 and 3 would not be doable outside of a hospital. Yeah.
Thank you so much. Now, regarding the 10,000 patients recruited so far for Recover, are you still working toward a total of 18,000? And do you consider the recruitment to be on schedule? And finally, what is the target date for completing that recruitment?
Right. Okay. Well, so we have certain bins of patients that we are trying to recruit. And so in terms of adults who have what we call post-acute sequelae of COVID, they're 6 months out, still have symptoms, we're pretty much done. We want to increase the diversity of the population, but that's our goal right now. We have hit our our kind of general number, but we still need better diversity. The area where we're still aggressively recruiting is in acutely infected people. So these are people who have the infection now and then can be followed to see how they recover. A certain proportion of them we think will make a good recovery. What we know so far is that there's a sizable proportion of people that have been enrolling in that study that are still having trouble at 3 months out. So clearly we have not hit the end of this problem. We're still recruiting people with acute infection, and they are still having trouble that looks like long COVID. So that area is still actively recruiting. Pregnant women recruitment is still active, and children is still active. So the answer to the question depends on, child, pregnant, not pregnant, acutely infected, or chronic problem.
Wonderful. Thank you very much. We have time for one more question. Let's end with this one. What is the most promising of the pathologies you spoke of?
Well, I think, it's early in the game and we're guessing still. So, but in fact, you have to make priorities. You have to start, and so you have to pick one. And the one, as I said, that's rising to the top looks to be this idea of persistent viral replication going on based on the autopsy study at NIH, which again is not published yet. It's still in the preprint form. But we've had discussion with those investigators. And then also the evidence of persistent viremia, not viremia, I'm sorry, persistent antigenemia in exosomes circulating in blood of people who have post-acute sequelae of COVID, that those two could go together. If you're having persistent virus, you're going to expect to see persistent antigens. But the caveat there is that they may not be tied together. You could have persistent antigens that's related to your infection months later, and it just takes a long time to leak out. So there's still another non-viral replication explanation for the antigenemia. But anyway, what we're going after first is this idea of trying antivirals for persistent viral replication.
Dr. Koroshetz, thank you for your time today. Would you like to provide any final comments for our audience before we go?
Well, I just think, people are kind of used to seeing COVID, and it doesn't look that severe as it did in the beginning with people going into intensive care units. But there's still this percentage of people, maybe 3 to 4%, who develop persistent symptoms. So, and that we know is reduced by vaccination. So I think it's another good reason to get vaccinated. And I guess my other comment is just, again, a big thank you to the tens of thousands of people working on this project. It's the biggest project I've been ever seen here at NIH.
Well, thank you again, Dr. Koroshetz, for your time today and for your important research. And before we go, I do want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until October 19th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everyone, take care and bye-bye.
Take care.